Hello everyone, good afternoon, and thank you for joining me today. I'm going to talk about achieving reproducibility. Within this topic, I'm going to give you a brief introduction about CST. Then we move to how serious the poor reproducibility situation is we are facing right now. What are the causes of irreproducibility? Who should be responsible? What is being done to address the problem worldwide right now? And then we come to the conclusion and question taking. CST is a private global company founded by a PhD scientist with a goal in mind for generating useful reagents that researchers could use to make clinical discovery. CST focuses on the application-driven antibody development and its validation. Also, we have put a lot of efforts on quality control and technical support. Internally, we have generated three generations of recombinant monoclonal antibody technology. That leads to generate recombinant rabbit monoclonal directly from polyclonal antibody. That is the latest technology. We have over 90% of recombinant rabbit recombinant antibody. We have almost 100% of manufacturing in-house. Now, is there a reproducibility crisis? Yes. Since 2012, scientists have raised a concern about science fraud and non-reproducibility. The initial analysis showing here, as you can see, only 11% of previously published scientific findings were confirmed. Only 28 of in-house results matched the published data. That's very serious issues. In 2016, Monia Baker from Nature have done a survey based on 1,576 participations, mainly coming from biology, medicine, and physics and engineering. They found more than 70% of researchers have tried and failed to reproduce other scientists' experiments. More than 50% of researchers have failed to reproduce their own experiments. That's a very large number of percentage. Further studies that done by Friedman and his team, they estimated over 50% of preclinical research was not reproducible. In other words, more than 28 million NIH funding given to academic research every year is wasted. So it is very serious. Although not clear how much antibody is involved, it seems biological reagents and materials were probably the largest fraction contributing to the waste. This is due to poorly identified reagent, poorly qualified reagent, bad reagent, reagent on wrong system that are made of a bulk of research wasting. Also, the investigator didn't set up the study properly, didn't adjust their proposal if the things is not what they expected, and didn't do or don't do biological replicates or repeats. Inappropriate data analysis, selective reports, for example, cherry-picking information, or the protocols used were not correctly recorded or incomplete. Therefore, such information being developed in the preclinical research will be wrong and incorrect, which will slowly and accumulatively lead to bad diagnostics and therapeutics. There has been happened in the history. Certainly, the lab protocols also include the misconduct, etc. I will talk about a little bit later. Now, if you compare the research use only antibody versus to diagnostic and therapeutic antibody, although it's a very small part, but they have a least regulation. As you can see here again, over 300 manufacturers generate worth $2.5 million of antibody. Unfortunately, there are no universally accepted guidelines or standard methods for determining the validity of these reagents. That's a very serious issue. Now, recent studies based on from Monica and the GBSI, they found the causes of a scientific irreproducibility. They believe these are the causes mainly caused irreproducibility, such as incomplete reporting, reagent variability, lack of validation, experimental bias, analytical techniques sitting on the top. For example, incomplete reporting means the selective report, cherry-picking information. Reagent variability means the lot-to-lot consistency issue. Lack of validation means the antibody didn't do enough validation, just put on the market to make money. Experimental bias comes up when people don't do biological replicates or repeats enough. Improper statistic methods used for statistic analysis. Lack of controls. When you do the experiments, you don't know what is the, if the system is working or not because you don't have the proper controls. Incorrect interpretation, which means you are subjective. You just always based on your proposal, just, even twist the interpretation, which is really dangerous. The misconduct can also cause the problem, although it's a very minor thing. It's not the major things in the most of the labs. However, we still need to pay attention to. But CST believes these causes can come down to three major problems. They are inadequate training and support. Little or no reagent identification means the reagent being used is not traceable, etc. Methods or protocols used were not completely correctly recorded. Reagents such as antibody were not validated enough, or antibody were not correctly used. Reproducibility can come down to the three things we call it: mentoring, methods, materials. We put into short 3M. Let's look at the training and support first. For the young investigators or fresh coming into lab postgraduate students, the lack of basic training. They have poor knowledge about antibody, how to use antibody properly. They don't have enough technical experience. Furthermore, they are not familiar with the value and don't see the need for the antibody validation, although the vendor should do antibody validation for you. But still, you need to do validation to a certain extent on your system, in your hand, to make sure it's going to work in your specific system. For example, the application-specific issue. A lot of people believe the performance in one application, for example, in the Western blot, it's going to work in the other application in the IHC, which is not the case. So that's why we strongly suggest if you use an antibody, a commercial antibody, you should follow vendors or manufacturers' suggested protocols, because that protocol is optimized. It's the best possible that you can get the best possible results. However, if you have any questions, don't be shy. You should go to the company or vendor to seek the technical support. They have the specific team to support you. They should play a role in the mentoring process. For the methods, we highly suggest the method should be clearly detailed, complete, black and white. It should be available, easy to access, let's say on the website, or should come in with the product. It should be repeatable and replicated. In terms of materials, for example, the antibody should be highly validated. What does it mean, the validation? I'll talk about a little bit later. And it should be easily identified, means it should be easily traced back and should be very consistent from lot to lot, and certainly should be used properly. How to deal with these three things? Who should be responsible? We would like to argue that principal investigators and institutions should be responsible for mentoring and the methods first. They should train young scientists how to set up proper experiments, how to adjust research proposal when it's necessary, why need to follow proper protocols. That's what I mentioned. If you use the commercial antibody or reagent, you should follow the suggested protocol, because that's the optimized condition to assist you to get the best possible results. And how to do the statistical analysis with the proper statistical tool, how to proper use of reagent in terms of your homemade reagent preparation, how to document correctly, clearly, and properly in terms of your protocols, summaries, interpretation, etc. On the other hand, scientists or people who are working just in front of the bench, most of the time, they use the reagents a lot. They should be able to clearly identify the materials, clearly document the protocols and the reagents being used to make sure other people can replicate their experiments. That's their responsibility. Whereas journals should be ensure they establish rules and guidelines to require correct citations, proper references for antibody being used in the paper. Vendor, including CT itself, should be responsible for all these three areas to make sure all materials and methods are carefully and clearly documented. All products to our best knowledge, best possible tools to validate and optimize the protocol, etc. And certainly, the lot-to-lot consistency should be paid attention to very much, should take it very seriously, and also should be able to provide the technical support to assist or mentor your experiment if it's necessary. So what the role does CST play? We have put a lot of efforts in the technical support, etc. As I say, CST is an application-driven antibody company. It means that all of the protocols shown on our website have been optimized for the application for that antibody. We even provide detailed protocols for application-specific manner. For example, if this is a frozen IHC, frozen sample for IHC, or paraffin formaldehyde, etc., fixed samples for IHC, etc. We share significant data showing a little change away from our suggested protocol could lead to catastrophic outcomes for experiments. For example, the 3% change to 4% formaldehyde in your fixation condition for IHC will give you totally different results. While we're putting a lot of efforts in validating products and optimize protocols, best possible results or outcomes in the field is not always the case. That is why our tech support would come in to help you, even assist you to troubleshooting. Also, when you raise an issue or request, you will receive a help assistance from a specific application experts who validate that antibody for that application. For example, if you have a question about IHC, you will talk to our IHC team. If you have flow cytometry issue, you will talk to our flow cytometry team, the experts who validate that antibody for that particular application. We also provide application guide through the website, tutorials, and on-site training for some complicated applications such as PTM scan proteomics. We certainly do a widely cooperation with investigators who are not only developing new methods, but also who are trying to use our product on the methods which we have not validated. Doing such, this would be benefit new customers from our previous customers' work. Quality consists of specificity, consistency, sensitivity, reliability, stability, and manufacturing. Among them, stability issue is very important. Monoclonal rabbit antibody, especially recombinant rabbit antibody, is very stable. However, before shipping out, we still store them at 37 degrees for 1 week to make sure their performance has no difference before and after such harsh condition. So that's why we say the recombinant rabbit monoclonal antibody is very, very stable. Now, this is a very typical data sheet, comes with a product. Also, you will be able to find through our website. On this, I highly suggest you guys to go through such kind of a data sheet. It will help you decide if this is the right antibody for the application you're after and you should purchase. In general, the data sheet only show approved application-related information. For example, here, this antibody can be used for IFC, Western blot, IF, etc. And species also tested, meaning that the antibody has passed the rigorous application validation process, the highest or very rigorous standards set up by CST itself. For those, again, for those species listed, the antigen sequencing used for the antibody in generation is 100% identical across the species without any assumption. So it is a very reliable antibody you can use for the different species if it's stated in our data sheet. In this data sheet, you should be able to find the conditions for application information such as dilution points, the buffers used for specific applications, etc. If the antibody can be used for Western blot, there's a default that we provide a quick piece of detail about experimental condition. For example, if the primary incubation should be with BSA or should be with milk, etc. This is a very typical CST validation principles. We validate our antibodies in terms of specificity, sensitivity, consistency, and methods we use, and technical support we are going to provide. I'm not going to go through the detail for all of them, just focus on two of them. One is the specificity and the other is consistent. Bear in mind this principle, let's have a quick look about CST antibody development process. We have two independent groups. One is called the development group, the other called the production group. A new antibody development or exploring started from the target selection, followed by a project design based on the application requirements, the species requirements. For example, we would like to develop antibody against PDL1. If we develop or find an antibody for WB or Western blot, it doesn't make sense at all because most of the time PDL should be tested with IHC mode, which is the immunohistochemistry. Then we design the peptide, immunize the animal, bleed, purified antibody. Then we do the first round application testing with IHC as the front. Certainly, IHC alone is not enough. We also do other methods. That means the multiple methods testing to validate that's the specificity. And then it goes to the serial molecular cloning to find the best possible clone, recombinant, sequencing recombinant, and we run a second application testing, again with IHC, including multiple methods to support the validation of the specificity. Then move to the final clone selection. After that, the third application testing done by development group. If it's passed, then it will hand over to the production group where the larger lot will be generated, and then it will go through the fourth application testing with IHC and other methods by the production group independently. If it passes their standards, then it moves to the official launch. So as you can see here, almost 50% of antibody project in the CST is failing during the process. Sharing this data with you just to trying to let you see that how serious about the validation, about the reliability of the products, we CST take care of it. Now, as I say, I like to talk a little bit about how we determine the specificity. This is the incomplete table that listed out the tools to our best knowledge, to the best available tools we use to determine the specificity, such as the positive and the negative cells used. For example, here we have an antibody against EpCAM. EpCAM is expressed in HT29 cells, but it's not expressed in the HeLa cells. As you can see, the green, which is a specific staining, whereas in the HeLa cell, there is no staining at all. Again, it doesn't mean we just do only one immunofluorescence. We will do multiple methods to validate the specificity of the antibody. Also, if the target is on the organelle or somewhere else, we also still use the IF to make sure if it's on the right subcellular location. Beside that, we also do the Western blot with the subcellular fractionations. We actually have the related protocols and reagents you can use if you are interested to make sure what you have seen on the IF is also specific on the Western blot in terms of the molecular weight and the specific band, etc. Now, treat cells with specific agonists, inhibitors, or growth condition to shift target moving around is also very important. For example, here, this is EGFR in A449 cells and HeLa cells. It's different. Let's say after treatment with EGF, the EGFR in these A549 cells will be shifting, whereas in the HeLa cells, there's a phosphorylation happen at the site of 1068. CST also compare our products against other companies or competitors. We would like to validate antibody, for example, here to see. This is an example that we have an antibody against prostatic acid phosphatase. The top one is ours. The second and the down one is the competitors. The purpose is to see if our product is better or worse or the same as competitors. If there's an improvement, we would like to put it on the market. As you can see, first of all, the Western blot, we show the same thing. In the IHC, it seems ours can give most strongest staining versus to the competitors. We also stain the cell pellets to pre-screen if the antibody could be used for immunohistochemistry. This is another example that CST anti-MET antibody on the left side are highlighted in blue and the non-CST antibodies on the right side. This is again positive and negative cells for a comparison. For example, here you can see the CST anti-MET antibody can clearly stain the positive cells, but there's no staining at all in the negative cells. However, the non-CST antibody can equivalently stain both negative and positive cells. In other words, there's a false signal in the negative cells, which is bad. Consistency is very important. That means the stability of the product. This is a very typical one of many examples that show consistency of CST antibody from lot to lot. It is a very classical golden product that generated at the early stage of CST establishment, which is the rapid polyclonal against phosphor P4442 ERK12. As you can see, the earliest lot generated in 1990, which is called lot 10. If you compare this band versus to the rest of lots, they are exactly the same. They are also in the same formulation and provide you with the same dilution point suggested. So this lot actually has been 17 years old, but it is still very good. So what is being done worldwide? Addressing reproducibility issue needs entire science community's participation. A number of initiatives have been proposed, and here is the proposed solutions. For example, reagent ID repositories. It is called RIDs, and the protocol sites, which is called protocol IO, now available on the website. You can go to the website and find out. It's very easily. And using this will be very useful to track down reagents protocol use in the publication, in the lab, and customers are encouraged to go to these two sites. It is a good way of citing, reviewing, and comparing protocols, reagents being used in the publications and in your work. There are a number of antibody research search engines. They provide the time of publications, number of citations, or end user data for that antibody used. It is very helpful in terms of which antibody are most likely used or highly recommended by others. Certainly, going to the vendor's website is the best, because that will give you comprehensive information data you want to see. Journals are slowly changing their policy to request better citations and a better user behavior by implementing checklists to make sure the authors correctly list antibody and protocols used. NIH has launched a program called Rigor and Reproducibility in Research in finding ways to greater reproducibility and accountability in research. CiteUp is a group based on UK. They launched a meeting early of 2016, bringing scientists, vendors, journals together talking about the antibody validation. It is now a very popular body. You will be able to get a lot of information about the quality of reagents, etc., specific antibodies. Most importantly, the global biological standards, in short, GBSI, recently held a validation workshop and reproducibility initiative by inviting leaders from research territory, journals, funding agencies, antibody database search repositories to talk about what's wrong with the antibody validation, what we need to do to fix it. Out of the meeting, these are the outcomes or suggestions we would like to implement. For journals specifically, they are going to link the reagent resources identification site into the paper, so people should be able to trace back what the reagent being used, the catalog, the reagent lot, the reagent lot, where antibody is coming from, the resources, etc. It will increase the transparency about this information. Vendor has been closely working with GBSI to adopt the validation standards. Specifically, they need to detail the protocols used in the experiments. They should provide the protocols in detail to the customers, which should be complete. And they also need to increase their transparency about the data, resources, application, validation about the reagents or antibodies being launched or put on the market. For the researchers, we highly suggest that the training mentoring is very important to make sure the young investigators being cultured from very beginning or baked from the very beginning in the right way, doing the right thing. Beside what we have gone through, I would say finding a good antibody is also important. I would say it's an initial step to reduce or minimize the irreproducibility. For example, again, if you do a Western blot and you just simply use the antibody for the application in terms of IHC or IF, you may be wrong, because again, one antibody works in one application does not mean it will work in another. So you should go through the data sheet or product insert detail before you decide to buy. On the other hand, for example, also I would like to mention you, if you want to do the chip assay, beside the signal versus to noise ratio, you also should pay attention to if the right target being involved in the validation. For the species reactivity, you should be looking at carefully if the species be mentioned in the data sheet or in the product insert, or that is the species or model you used or you are going to study. Protocol antibody application specific, again, like what I mentioned, if it works in the Western blot doesn't mean it's going to be working in IHC or IF. If the protocol optimized, this is another important thing. A lot of vendors or some vendors or companies just provide you, oh, this is the concentration for this particular antibody. You should decide the optimal dilution point. It doesn't make sense. They should give you as narrow range as possible in terms of a dilution. For example, we give you the dilution point. Certainly, you need to do just a little bit, validation, lower validation and higher validation. You do the pilot study, then you decide. Because like what I say earlier, it is not always the case that the best possible results coming out in the field versus to the in-house. There's a little bit difference. Also, you need to know if what is the type of or the clone of the antibody you are using, if it's recombinant or if it is the monoclonal or polyclonal. If the polyclonal is the only available product in the market, you should be careful. You should be check out if there is any dilution points change from the different protocols, because that will indicate the consistency of the lot to lot. It is also important to take peer review about the citation of the antibody, which will tell you which antibody has been highly recommended and used in the other labs. If the company can give you the best possible technical support, that's another important thing you should pay attention to. You should be aware of that. Reviewing with the citation, etc., you should be very cautious, because that's a simplified words. It's not going to give you the detail. It is better to trace back to the original paper. In fact, CST sponsored webinar antibody validation roundtable in 2014 by inviting people from journal, from research area, from diagnostic manufacturing, antibody research use only manufacturers. They discuss one of the point is that, and one of the important things they discuss is that a good antibody should be qualified with proving specificity, proving sensitivity, proving specificity in the application, and proving reproducibility. Having said that, now it's our turn being open-minded. What is your definition of reproducibility? What does a researcher or you need to do to ensure your experiment conforms with acceptable standards? Why the validation is important in terms of, the antibody's validation is so important. What factors play a role in ensuring the reproducibility of experiment results? We hope we have a reproducibility roundtable discussion in the near future with Korean researchers. CST and BRIC will lead this in the future. We like to do so. We would like to work together with you. Last but not the least, it isn't always just the antibody cost issue. For example, the recommended protocol is not followed. Again, the antibody should only be used for Western blot, but you would like to try IHC, it's fine, but you need to do a pilot study to figure out, or you go to the company, seek help, assistance, raise your question, inquire the technical support. Using application that is not recommended, supported, it's similar to the first point. The other thing is bear in mind is the poor sample quality or poor sample preparation. Also, the reagent preparation is not right, or the instrument, something's wrong, and the instrument hasn't been calibrated or normalized correctly before you use. All of these could cause problems. We have distributors in South Korea. We have application sales manager in South Korea. We have marketing manager for Asia Pacific in South Korea, basically. The most important thing, we have application specific technical support from scientists in CST who are validate antibody in that specific applications. So if you have any questions, come to our distributor, our application support in your country, or go to our Boston-based company's technical support. We work together to make it happen. Thank you. I would like to take a question. Thank you.